Okay, so this is going to be the video covering the fluid mosaic model of the membrane, chapter 11. And so some of the take-home messages that we want to cover are why we're going to get spherical bilayers, the energetic favorability of having a bilayer and having it be spherical. Some of the components of the membrane, it's more than you would think, including besides phospholipids, um, there are also cholesterols and sphingolipids and lots and lots and lots of proteins. We want to talk a little bit about the fluidity side to side, but that there's very little fluidity from inner leaflet to outer leaflet. We're going to talk a little bit about the cortex uh, lying on the inside of the membrane, and then we're going to talk a little bit about glycosylation or adding sugars to proteins and when and where that happens. So, so the fluid mosaic model is uh, that the membrane has fluidity that things are actually just floating in it, um, the membrane, that the membrane structure is not static, but instead very, very dynamic. The, the phospholipid bilayer includes many other things besides just the phospholipids. We do have the phospholipids, the phospho head group attached to a couple of lipids, and we're going to have that bilayer, so it's going to go phospholipid, lipid, phospho. There are also cholesterols that are kind of riveting those phosphates, I'm sorry, the lipids together, um, creating a more consistent fluidity. There are going to be transmembrane proteins in the phospholipid bilayer. Most of these proteins um, will have large sugars attached to them. So chains of carbohydrates, individual sugars that are linked up to each other and attached to the protein. They're going to be attached to either nitrogens or oxygens on R groups. So there's N glycosylation and O glycosylation. These large sugars can also be attached to lipids, so we can have glycolipids or glycoproteins. There's a lot of proteins, filamentous proteins, attached on the inside, the inner leaflet of the cell. So on the cytosolic side, these are going to include cytoskeletal proteins as well as proteins that are linking them together. There are proteins that are attached to the integral membrane proteins, and we call those peripheral proteins. So the phospholipid bilayer under EM has a kind of a trilaminar look to it, and that's because we've got the phosphos being electronically dense, um, and so the EM does not go through the phosphos, and so we'll have a dark stripe, and then a white stripe where the lipids are because they're not as dense, and then another black stripe. So it's going to go phospho, lipid, lipid, phospho, and that's going to create this trilaminar. So there's going to be water on the outside of the cell, water on the inside of the cell, and then the phospho, lipid, lipid, phospho, creating the bilayer. The phosphos and the head group are highly charged and therefore hydrophilic, and so they're going to interact with water, whereas the hydrophobic tails are going to be sequestered away from the water and only interacting with each other. And this is what creates the need for a bilayer. There is a way to sequester lipids away from water in a single layer. That's called a lipid micelle, and it's not too hard to create lipid micelles. You just strain phospholipids through a very small hole into water, and you're going to create these very tiny, maybe 50 to 60 microns in diameter lipid micelles. But there's going to have to be no water on the inside of that micelle. And so because there's no water on the inside, this is not a very good plan for a cell. It's just too small to contain the genetic material and then all the proteins that are responsible for intermediate metabolism. So the phospholipid bilayer is, like I said, made of phospholipids. That phospholipids always have these hydrophobic tails attached to a glycerin. So there's going to be two of them, and so this would be a biglyceride or a diglyceride. And then attached to that will be a phosphate head, and then attached to that will be some sort of head group. These head groups are either serine, choline, ethanolamine. There's several head groups that they could have, including, instead of a phospho and a head group, you could have a large sugar. So you could have a glycolipid, so a large chain of glycose attached to an O on that glycerol. And then, like I said, there was also going to be these steroids, these cholesterols that are creating a more consistent fluidity, more stability of the phospholipid bilayer. So both inside and outside of the cell, there's going to be water, and that creates that need for the sequestering away from hydrophobic lipid, and the need for that polar head group. Sugars are polar, and the phospho is polar, and the head group is polar, and so those are going to interact with the water and create the phospholipid bilayer. So they are naturally going to attach to each other because the lipids are going to want to stay away from the water, and the best way to do that is to attach to other lipids. And so you can easily get these large sheets, but the problem with these large sheets is that all along the edge of the sheet there's going to be exposed lipids. And the best way to create a structure that has no exposed lipids is for that planar phospholipid bilayer to wrap around so that these lipids are interacting with these lipids, and these lipids are interacting with these lipids. And so the energetically favorable conformation is going to be a spherical bilayer, and then you won't have any lipids exposed to water, and only the polar things will be exposed to water. Like I said, there's going to be more than one kind of head group, and in this case, the inner leaflet and the outer leaflet are not identical. The outer leaflet, the one that's exposed to the extracellular space, is going to be rich in sphingomyelin and phosphatidylcholine, and the glycolipids, whereas the inner leaflet is going to be much more rich in phosphatidylserine and phosphatidylethanolamine and phosphatidylinositol. Phosphatidylinositol is on the inside because it's important for signaling. Phosphatidylserine is also a signal, but it's a signal for cell death or cell suicide. If there are ever any phosphatidylserines in the outer leaflet, then that is a signal to start apoptosis. And so, we actually have proteins that can move phosphatidylserines inward and the phosphatidylcholines outward. And those proteins are actually called flippase and floppase. Flippases move things to the inner leaflet and floppases move things to the outer leaflet. If there's ever a signal for apoptosis, then you get the production of a protein called scramblase, and scramblase can move the uh, phospholipids in either direction. And so then you'll start to have phosphatidylserines on the outer leaflet, and that's a signal to macrophages to engulf the cell. So we keep the phosphatidylserines and the phosphatidylethanolamines on the inner leaflet using flippases, and we keep the phosphatidylcholine in the outer leaflet and the sphingomyelin in the outer leaflet for the most part using floppases. So there are more flippases than floppases, and so it's okay if there's some sphingomyelin or phosphatidylserine in, I'm sorry, phosphatidylcholine on the inner. That's not going to cause a problem. So another thing to remember is that the cytosolic side always stays cytosolic. So these phospholipid bilayers start off in the smooth ER. The ER is the place where these things are produced, and then when you package proteins for them to go on to, say, like the Golgi, or when they bud off of the Golgi and go to, say, the lysosome or the plasma membrane of the cell, they're always going to bud off, and then when they meet each other, they're always going to um, uh, the the vesicle will fuse with the target membrane, and so the dynamics of that causes the cytosolic membrane to always stay cytosolic, and the luminal membrane, if it goes to the cell surface, would be the extracellular membrane leaflet. So there's the asymmetry is always preserved by the dynamics of the budding and fusing. So so flippases, floppases, and scramblases. These are these are uh, active transport. This is going to require ATP to allow for this to happen. And so, like I said, flippases are going to move things into the inner leaflet. So the way I remember that is flip has an I in it and inner has an I. And floppases are going to move things to the outer leaflet. And again, flop has an O and outer has an O. And scramblases, they can go in either direction. So there's not much movement in to out that requires energy, but moving side to side, that requires no energy. We're going to investigate that a little bit more in the next video. Okay, so move on if you want to.